Alright, so once we have these two strands that were in the helix unwound, then they're no longer connected by double bonds, and we can actually have free nucleotides coming in and being added on. So, for example, if we've got one strand of our DNA here, then each of the nucleotides along here can actually be paired with. So we've got our free strand, which came from our original parent DNA. So this is the parent. So what we say is this is the template. So the template can actually start creating a new strand. So what we have is what we call our nucleotides floating around. So the nucleotides will be one of our four bases attached to the sugar and the phosphate. So we've got all of our different DNA nucleotides floating around in solution, and what they can actually come and do is start pairing with our parent strand. So by doing this, essentially what we're doing is we are making a new DNA strand. So this is how we actually make our new strand, which is exactly complementary to the other strand that the strand was pairing with in the DNA double helix. So we do exactly the same for both strands of the original parent. So in this way, we're actually creating two new DNA helices. So I'll show you how this sort of works as a representation. Alright, so we've got our original parent, which contains two strands. So we got those two strands, and the first thing is we separate those strands. So we separate them through the helicase, which unwinds it. And then we start the process of the replication, where as well as having that each of those individual strands, we now have created these new strands to pair up with those bases again. And by doing this, we're recreating the double-stranded helix. So here's the place where we're having the DNA replication. So we can see from that one parent strand, we've actually created these two new helices, which are going to be, one is going to go into one daughter cell and one is going to go into the other daughter cell. So that's how we end up with our two double-stranded helices. It's the process of unwinding our original parent, then using each of those strands as a template to make a new strand which completes the helix again, and then by doing this with the two different parent templates, we've got our two new DNA helices within that cell, and then we can divide through mitosis, and one of these helices can go into one cell and one can go into the other. Remember that it's not just going to be a single DNA helix that is going to be replicated. There'll be huge numbers of these to copy all of the DNA content within the cell. So we're just using the example of one single helix just to simplify it. So we have the enzyme called DNA polymerase, which is actually the one that is responsible for joining these bases that we've added onto our new strands. So I showed you up here how we come in and add these new nucleotides. So what it actually does is it connects these nucleotides and also will check that the sequence is correct. So we need to make sure this sequence is correct, otherwise there will be defects in the DNA that we've copied, or what we'll see later called mutations. So what we're actually doing when we're connecting these together, we're creating those phosphodiester bonds between the sugar phosphate backbone to connect the nucleotides. So we saw in the earlier slide that we have the sugar phosphate backbone connecting through individual nucleotides on the same strand, and then the side chains actually form hydrogen bonds to the nitrogen-containing bases on the other strand, and that's what gives the DNA helix its structure. Alright, so when we consider DNA replication, one of our strands runs from 5' to 3', and one actually runs from 3' to 5'. Remember, they're anti-parallel. So the one that runs from 5' to 3' is what we call the leading strand. So it's the leading strand because it can be replicated more quickly, whereas the lagging strand is replicated more slowly because it makes it more difficult to actually do it because of the direction it's in. So we'll see this a little bit later on. So at the top here, our old strand is actually running from 5' to 3'. So this makes it the leading strand. Whereas down the bottom here, we're actually running from 3' to 5', which makes this the lagging strand. So let's have a look at why this matters. So because the leading strand is 5' to 3', we're actually able to create a new strand by growing in the 5' to 3' direction. And because our enzyme, DNA polymerase, can function only in the 5' to 3' direction, we can actually make the leading strand quickly. So we can do this quickly because we're just adding in those new nucleotides and joining them up through the DNA polymerase. So we can do this nice and quickly. There's nothing stopping us from doing this. Whereas in the lagging strand actually runs in the opposite direction, 3' to 5'. So that means when we're trying to make a template, we actually can't grow continuously. Because DNA polymerase only works in the 5' to 3' direction, we can't make a fragment continuously from our 3' to 5' lagging strand. Instead, we use what we call Okazaki fragments. So our Okazaki fragments are actually short nucleotide sets which are made in the 5' to 3' direction. So this is made with DNA polymerase, and we can actually get these to match up to our lagging strand. And then when we get multiple of these Okazaki fragments together, we can connect them through DNA ligase. So the lagging strand is not continuous. We have to make the new strand in short fragments, whereas the leading strand can actually be made quickly and there's no delay. So that's the difference between the leading strand and the lagging strand. So when we finally have made our new DNA, we can see here what's happened. The parent originally had 2 strands. We then separate those through unwinding the helix, and then we make our new strand through this DNA replication. So each new DNA molecule that we've created, or DNA helix, contains 1 new strand and 1 old strand. So you can say it's 1 strand from the original parent and 1 newly created strand. So let's visualize our different types of replication here. So the top here we have our leading strand, which itself runs from 5' to 3'. So 5' at this end, and all the way down here it's at 3'. So that means when we're trying to create a new strand, we can actually use DNA polymerase to make this very quickly, and this is by making the strand by going this way, and in that way we're going 5' to 3'. So we can do this nice and quickly, and it's nice and easy to do this. So we have this continuous growth. However, for our bottom strand here, we actually run from 3' to 5'. So this is our lagging strand. So when we're trying to create a new DNA sequence, we can't actually grow continuously from right to left, because this would be going from 3' to 5', and we can't actually do this because DNA polymerase does not work this way. So instead, what we actually have to do is make short segments that match up to our lagging strand, and these are our Okazaki fragments. So the Okazaki fragments will complement the DNA, and when we've got all of these added in, we can connect them through the DNA ligase. So you can see this is not continuous growth. We call this growing in segments, and the segments that we're using to grow here are the Okazaki fragments. So you can see how the lagging strand is growing slower, whereas the leading strand will grow more quickly. And it's all about the direction of growth as either 5' to 3' or 3' to 5'. So if it's 5' to 3', we can do it quickly. If it's 3' to 5', as it is with the lagging strand, we can do it more slowly. So why is it that the leading strand is able to grow so quickly? This is because we can grow the new strand continuously from 5' to 3' using DNA polymerase. Why is it that the lagging strand is much slower? This is because we can't actually grow continuously in the 3' to 5' region, so instead we have to make short Okazaki fragments to match up with the DNA sequence in the 5' to 3' direction. So another important point about our DNA replication is the fact that we actually have multiple replication sites within our DNA. It's not just one replication fork opening up, allowing us to start copying the separated strands. We actually have multiple points, which we can see in this next slide. So we're actually opening the strands up in multiple locations here, so we can actually start replicating the DNA in multiple points, which allows us to eventually get to the situation at the end where we have those two new double-stranded DNA helices at the bottom here. So by opening up these multiple points or multiple replication forks, it means we can actually start the replication process at more positions, and eventually they'll all just join up. So it's more quick than just opening up one replication fork and then copying from that position. So one thing that our chemotherapy drugs actually do is they actually try to interfere with DNA replication in cancer cells to cause them to actually die. So on the surface, this looks like a good way to treat it because we know that our cancer cells grow very quickly, and if we can stop them growing, then they're going to die. However, the problem with the chemotherapy drugs is it actually targets a lot of other cells within our body that grow very quickly. So this is why someone with chemotherapy treatment will be having quite a low white blood cell count. So you can see here a really nice summary about DNA replication process. So the first step is to unwind our DNA. So we are separating our two DNA strands. So we have to separate the DNA helix, and to do this we use our enzyme called the helicase, DNA helicase. So once we've done this, we can actually get those free nucleotides coming in and binding to the old DNA strand, and then we can get these being connected through our DNA polymerase. However, we can only do this from the 5' to 3' direction on the leading strand, whereas on the lagging strand we actually have to make short segments called Okazaki fragments because we can't grow continuously due to the other strand running from 3' to 5', and this is what we call the lagging strand. And then the last thing that we'll see is we've completed our DNA replication. We're going to have those two new DNA helices, and one of those strands is from the original parent and one is a new strand. So each one contains one parent strand and one new strand. Alright, so looking at some of the differences between our DNA and RNA, so we had a look at a few of these earlier. So we saw that the sugar within our nucleotide differs between our DNA and our RNA. So in DNA, we have the deoxyribose, deoxy because it has less oxygen, and the ribose is one of our monosaccharides with 5 carbons. So they both have 5 carbons and are monosaccharides, but the difference is that we have less oxygen in the deoxyribose than the ribose at the carbon 2 position. We also had a look at the nitrogen containing bases, so our adenine, thymine, these types of things. So in DNA, we have adenine pairing with thymine by 2 H bonds, whereas cytosine and guanine actually form 3 hydrogen bonds. But in RNA, we actually have uracil instead of thymine, so adenine pairs with uracil by those 2 H bonds. And again, we have the cytosine pairing with the guanine by using these 3 hydrogen bonds. As we've seen, the DNA mostly is seen in these double-stranded DNA helices. And we'll talk a little bit now about our RNA, but mostly we see the RNA as a single-stranded species, or at least it will be in the lecture today. So the other big difference between our DNA and our RNA is the size. So the DNA is very large, so huge number of nucleotides together, up to 1 million nucleotides, whereas the RNA is actually much shorter because we're just copying a specific region of the DNA that we need to make a protein. So although our RNA is just made up of a single strand, we can have it forming what we call secondary structure. So remember, secondary structure is how something arranges in space. And these can be arranged in space differently because they actually have the capacity to form hydrogen bonds together. So just like in our DNA helix, the 2 strands were able to form hydrogen bonds between their nitrogen-containing bases, the RNA can also form bonds between the nitrogen-containing bases, but the difference here is that there's only 1 strand. So what actually happens is the strand actually folds back on itself to allow this pairing to happen. And we have what we call a hairpin loop at the end here, where there are no base pairs, and this is due to the bases not being able to be close enough together, or the pairs don't actually match up. So we can see how this structure here is quite different to if there was no pairing at all. So if there's no base pairing, we would just see this as a straight line. Whereas because we get this pairing, the RNA is actually able to loop back on itself and stabilize that structure by forming these interactions which basically hold it in place.